The thick filaments inside of a sarcomere are made out of many myosin proteins all bundled together. When we look at myosin as a molecule, it has a very unique three-dimensional shape. It has this linear region here that's referred to as the coiled region, two chains coiled around each other. And then it has this region at one end that is referred to as the globular heads. Now there's some other language used to describe the myosin molecule in this diagram. Don't worry about this language. Just understand that part of the myosin molecule is composed of two coiled chains, and there are two globular heads at one end that protrude off of the protein. These globular heads are especially important in the function of myosin in the thick filament. Now, myosin is found not only in skeletal muscle, but also in cardiac and smooth muscle. It's even found in some non-muscle cells. It's a motor protein. In other words, it is capable of physically moving, changing location under the influence of the hydrolysis or the breakdown of ATP. So with the addition of some energy, this protein can change shape and cause physical movement. And that movement is what allows for contraction to occur. So part of the myosin molecule, specifically the globular heads, has catalytic activity. It acts like an enzyme. It can bind ATP and it can break off the outer phosphate group, taking ATP to ADP and releasing a phosphate group. That's also going to release a little bundle of energy. That energy is then used for the mechanical force needed to shorten the sarcomere and contract the muscle. During the contraction of a sarcomere, the myosin heads are going to attach to and pull the thin filaments. This is where the arrangement of the thick filament really comes into play. The mirror image with the heads at either end is going to allow for those heads to bind to hold on to the thin filaments and pull them towards each other. That's going to cause the sarcomere to shorten. Actin, thin filaments, is actually sliding past the myosin as the myosin heads move, and that's what creates contraction. Now, we said that thin filaments are made out of many copies of a protein called actin. Those actin proteins are not bundled together the way that myosin proteins are to make a thick filament. Instead, the actins are joined together into a strand, and two strands of actins are going to wrap around each other to create this structure called a thin filament. Again, thin filaments are arranged around the thick filaments in a hexagonal pattern. And when the myosin heads start the contraction process, they're going to bind to those thin filaments and pull them so that the thin filaments slide past the thick filaments. Now, we said that there were additional proteins involved in the sarcomere, and two of those proteins are associated with the thin filament specifically. Those proteins are called troponin and tropomyosin. On this slide, down at the bottom, you can see a drawing of a thin filament. Each one of the blue circles represents an individual molecule of actin, And you can see that there are actually two strands of many actin molecules all joined together. The two strands are wrapped around each other to create this filament. Now, in addition, there's also this green molecule. Kind of looks like a red vine, a stick of licorice. And you can see there's one here, and then there's another one here. These are proteins, and this is called tropomyosin. So the tropomyosins lie sort of along the length of the thin filament, and they're spread out at intervals. Now there's a second protein that's much harder to see in this drawing. It's located right here. It's a protein called troponin, and troponin is actually composed of three different parts. What's called troponin I, troponin C, and troponin T. Now we shorthand that as TNI, TNC, and TNT. The I and the C and the T are there to explain which structure these three components bind to. So both troponin and tropomyosin are proteins, and they are closely associated with the two strands of actin that make up this thin filament. Now, if you look at the image on top of the slide, you can see a drawing of a sarcomere. Here is that M line that we see in the middle of the H zone. Over here, this is a Z line. Now they're calling it a Z disc. Don't worry about that language. Here's the other Z line. And in between those two Z lines, we have a sarcomere. The yellow bars represent thick filaments. The brown bars represent thin filaments. And you can just see there's a little licorice stick structure right here. That represents tropomyosin. And then there is this blue ball right here. That represents troponin. So troponin and tropomyosin are critical to muscle contraction because they're going to play a role in when thick filaments can come in contact with thin filaments. Tropomyosin, again, is the elongated one, the long licorice stick looking twisted rod of a protein. And tropomyosin, again, lies in the same direction that the thin filament lies. In fact, because the thin filament is made out of two strands wrapped around each other, there's a groove between those two strands. And tropomyosin lies really close to that groove. It's not actually in the groove, but it's close to it. The groove is important because it's in the groove between the two strands of actin that the binding sites are found for those myosin heads. Remember, the myosin heads have to bind to the thin filaments in order to pull them. So there are binding sites on thin filaments, and those binding sites are found in the groove between the two strands of actin. Now remember, troponin, we said, has three individual parts to it. It's three proteins that function together as a unit. The TNT part of it, the T stands for tropomyosin. So troponin is bound to tropomyosin. The TNI region binds to actin, so the troponin molecule is also bound to the actin in the thin filament. It's bound to the actin, it's bound to the tropomyosin, it's really anchored. The third region in troponin, the TNC part, binds to calcium. So when calcium is released, and we're going to talk about when that occurs, but when calcium is released, remember, from the sarcoplasmic reticulum in a muscle fiber. That calcium is going to bind to the TNC region of troponin, and it's going to cause something to happen. All skeletal muscle fibers contain myofibrils, and those myofibrils are full of these sarcomeres. The sarcomeres are going to shorten. They're going to physically shorten during muscle contraction and generate the force, the mechanical force of muscle contraction. But it turns out that if you go into a muscle and you go down through the fascicles and into the different types of muscle fibers, it turns out that different muscle cells, different muscle fibers, can generate a different type of contraction. So there are different types of muscle fibers. Some of them generate contraction differently than others. Now, those differences between the muscle fibers are going to be based on the type of contraction, and that by that we mean how powerful it is and how fast it is, and also by where the ATP is coming from to power that contraction. So inside each muscle fiber, the ATP is required by the myosin, remember. It's going to bind to the myosin, and the myosin is capable of breaking off the outer phosphate group from the ATP and releasing the energy needed for that mechanical force of contraction. The place where that ATP comes from, the process that makes that ATP, is going to vary from muscle fiber to muscle fiber. So let's take a look. Let's start with differences among muscle fibers based on the type of contraction that's generated. By type of contraction, we mean how powerful it is and how fast it is. Now, the power of a contraction is related to the diameter of the muscle fiber. Larger muscles, again, grossly visible muscles, have a greater diameter across them. And inside that muscle, inside the fascicles, when you get down to the level of the cells, the muscle fibers, each individual muscle fiber can have either a smaller diameter or a larger diameter. The muscle fibers that have a larger diameter have more myofibrils in them. Remember, each muscle fiber is full of myofibrils. So if the muscle fiber is bigger, it has a broader diameter, it means it has more myofibrils in it. If it's got more myofibrils in it, it's got more sarcomeres in it. And if it's got more sarcomeres in it, it can generate more powerful contractions. More sarcomeres, more power. The other way we base type of contraction on, the other thing we base it on, is speed. Now, the speed of contraction is actually related to the myosins, and it's a genetic thing. Individual people, have genetic variations in their myosin molecules, specifically in the part of the myosin protein that has the enzyme activity that can split ATP. In some people, their myosin can do that splitting faster than in other people. So, using power and speed, we can describe two types of muscle contraction. We can talk about fast twitch and slow twitch. In a fast twitch muscle fiber, you're going to see a faster initiation of contraction, and you're going to see a shorter duration of the contraction. And I have an actual time down here. about 7.5 milliseconds. That's how long the contraction is going to last in the fiber. Now, slow twitch fibers, in contrast, are going to initiate the contraction more slowly, but they can hold the contraction longer, up to about 100 milliseconds. All right, so fast twitch and slow twitch. Now, years ago, when we were first learning the physiology of muscle, these were the only two types of muscle fibers that we described. We talked about fast twitch and slow twitch fibers, but those were the only differences that we really appreciated between different muscle fibers. But now we've added some additional differences to the types of muscle fibers that we see. And again, that relates to where the ATP is coming from that powers the myosin in the sarcomeres. So some muscle fibers get their ATP for muscle contraction primarily through aerobic cellular respiration. That makes sense, right? Remember, aerobic cellular respiration, the aerobic word means that the final electron acceptor at the end of the electron transport chain is oxygen. This is a very, very efficient way to make ATP, but it's relatively slow. It takes time, in other words, to get all the way down through this long string of chemical reactions. So there are oxidative fibers, and we also refer to these as red muscle fibers. They get their ATP for muscle contraction primarily through aerobic cellular respiration. Oxidative muscle fibers have an extensive blood supply. They have lots of that protein called myoglobin. That's what actually gives them their red color. They have lots and lots of mitochondria. And they resist fatigue better than other muscle fibers do. Now, let's think about this. The extensive blood supply, that's important because they need oxygen to do their aerobic version of cellular respiration. They need all those mitochondria because that's where all this cellular respiration is going on. So the anatomy of these fibers makes a lot of sense. But there are other types of muscle fibers. There are what we call white fibers, and we also call those glycolytic fibers that are getting their ATP a little bit differently for contraction, more inefficiently, but faster. And it's occurring through glycolysis. Remember, glycolysis, the process where glucose is broken down into pyruvate. Glycolysis is a process that does not require oxygen. So these fibers need less blood. They don't have to store all that oxygen, so they don't need as much myoglobin. That's why they look whiter or paler. They have fewer mitochondria. They don't need to do the whole process of cellular respiration and oxidative phosphorylation, but they fatigue a little faster. So two very different ways of getting their ATP for muscle contraction. So now when we describe different kinds of muscle fibers, we combine both power speed classification and where the ATP is coming from classification. So we combine both the fast and slow twitch language with the oxidative glycolytic language. And these are the classifications that we use. There are slow oxidative fibers. Those are also referred to as type 1 fibers. There are fast oxidative fibers. Those are also called type 2A fibers. And then there are fast glycolytic fibers. Those are called type 2B. Now you'll also see them called type 2X in some source materials. I have seen 2B used more often. So type 1, type 2A, type 2B. Slow twitch, oxidative. Fast twitch, oxidative. Fast twitch, glycolytic. 3 combinations of traits that you can find in different muscle fibers. Now you can imagine that what combination of muscle fibers a person has in their muscles would really impact their athletic ability, right? It turns out that most of the muscles in the human body contain a mix of those 3 types of fibers. But the percentage of each type of fiber often reflects the job that the muscle does. So think about, for example, the muscles that surround your eyes, the muscles that are in your hands. Think about the work that those kinds of muscles have to do. Those muscles have to be able to respond very quickly, but they really only need to contract briefly. So it makes sense that those kinds of muscles would have a high percentage of the fast glycolytic type of fiber. Now, in comparison, something like the muscles in your back, the muscles in your calves, those muscles are there for kind of continuous contraction, right? Your back muscles, your calf muscles, they are working all day, every day. So they're in need of slower but more continuous contraction. So when you look into those muscles, you're going to find the highest percentage of slow, oxidative types of fibers. So we will see the percentage distribution of fiber type based on the work that the muscle has to do. And we also see the percentage distribution based on the individual. People who are quite athletic have different distribution of muscle fiber type based on the kinds of sport that they're engaged in. For example, we tend to find more slow oxidative fibers in the legs of elite distance runners. Those people are performing in a way that requires the same kinds of continuous contracting work that you would find in the back muscles and the calf muscles. Slow oxidative fibers are the most abundant type. If you look at elite sprinters, though, or you look at somebody like a professional level weightlifter, they're going to have more of the fast glycolytic type because they're asking for a faster, a faster contraction that can be briefer in its duration. Now, something else to note, and I think this is very interesting, is that there's a lot of genetics involved in what proportion, what percentage of each type of muscle fiber a person has. And because of the genetic contribution, in other words, because of the fact that you inherit the percentages of each type of muscle fiber, that means that some people simply cannot become elite distance runners. Some people simply cannot become elite sprinters or Olympic level weightlifters because they weren't born with the right distribution. So you can bring about some changes through conditioning, that's been shown, but you cannot train your way, you cannot condition your way to these elite levels of sport unless you were also born with the right distribution of fibers. Now, interestingly, research has shown that through conditioning, if you train correctly and hard enough, some fast glycolytic fibers can temporarily, at least, while you're training, while you're working, in other words, while you're at that elite level, can temporarily begin to function as fast oxidative fibers. So you can improve certain certain percentages of the muscle fiber types for your sport, but a lot of it is based upon what you inherited from your ancestors.